Hello everyone, welcome to this webinar on file formats. My name is Michel and I'm a developer here at SVI, and the aim of this webinar is to describe how microscope images are stored after acquisition and to teach you about the differences in file formats to make sure you get the most out of your data for deconvolution and analysis. I will cover topics like bit depth and dynamic range, file compression and conversion, but I also would like to take this moment to stress the importance of saving your microscope's metadata, as this can be vital in performing reproducible deconvolution and quantitative analysis. If at any time during this webinar you have questions or you feel that something is not clearly explained, feel free to send us an email to info@svi.nl and we will get back to you in 24 hours. With that out of the way, let's dive into the file formats. First, let's start with some basics. After you have prepared your sample and have loaded it into the microscope, the acquisition starts. In this example, we see a confocal microscope. The excitation laser in blue is guided to excite a tiny 3D volume of your object that is in focus. This object is tagged with a fluorescent dye that re-emits light in another wavelength, shown in green, and this emitted light is then captured by your microscope's detector, where out-of-focus light is rejected by the pinhole, as seen on the right of this diagram. As the excitation laser is moved across the sample, the detector keeps track of the intensity it captured at each specific point and writes that away to a file. When imaging multiple fluorophores, the same points are sampled again, with a possibly different excitation and emission wavelength, and the process is repeated. The file that your microscope then saves after the acquisition is a collection of these different intensities for all sampled points as collected by your microscope's detector. Opening an image in Huygens can be as simple as dragging and dropping the image file into the Huygens main window. Every file has a file name followed by a dot and an extension that is often 3 letters at the end. This extension tells you what kind of file it is. Huygens supports the reading and writing of many different common file formats, but it also reads more proprietary microscope file formats from many different microscope manufacturers. Many of you are probably familiar with 2D image formats such as TIFFs or JPEGs, which are often still used to save image from, say, your smartphone camera, and these are also widely used online. They can also have a place in microscopy if used properly. But what makes these microscope file formats different from the JPEGs that your smartphone makes? Well, your smartphone mostly stores images in its red, green and blue components, whereas your microscope images contain intensity images of specific fluorophores. Both of them sometimes save storage space by compressing the data into smaller blocks, something we will discuss later, and thus saving precious space on your hard drives. Smartphone pictures can have metadata about the location where the image was taken, while microscope file formats often store the information to do with the acquisition, such as the laser wavelength you used, the information of the objective lens that was active during acquisition, and some acquisition softwares can even write tiled series of images for stitching, which requires information about the position of the microscopic stage. If you zoom in on images, you can see that the image becomes very blocky and you can distinguish the individual pixels. Each pixel represents a position where the microscope sampled your object. A higher intensity value correlates with more emitted light from your fluorophore that entered the microscope's detector. These are commonly visualized with gray levels between 0 and 255. This means that you have 256 different gray values for displaying purposes to distinguish objects by brightness and contrast. Here, 0 is completely black and 255 is completely white. The dynamic range is limited by your camera or detector. However, modern cameras often have a larger dynamic range, allowing for values well above 255. But in this example, we have 255. File formats also need to be able to handle larger values than 255 as well, and we'll return to this later. Make sure that during acquisition you try to do your best not to oversaturate the camera detector. It can only detect intensities between certain intensity levels. Larger signals above its limit can saturate the sensors and might not be accurately recorded. This is called clipping, and you effectively lose contrast information. Sometimes the signal of a fluorophore can look very dim and you don't see it that well. A solution is to increase the gain of your detector. That way signals get boosted. But this will also increase the noise in your image and comes with a real chance of clipping your images by oversaturating the detector if during the acquisition you move to a brighter part of the 3D volume. Be aware that even very dim signals can be improved with deconvolution by moving the out of focus light back towards your object. If you're interested in this, take a look at our previous webinar on deconvolution of low signal images. It is important to make a distinction between two types of images: RGB images and microscope images of multiple fluorophores. As mentioned before, a smartphone camera often captures colored images by capturing information specifically of the red, green and blue parts of the light spectrum. Then it combines this into an RGB image. A fluorescence microscope uses multiple fluorophores or multiple channels, and here we are not as interested in how the summed intensities of all fluorophores add together break down into specifically red, green and blue components. We are mostly interested in the total intensity of one fluorophore compared to the total intensity of another one. Both of these images look really nice, of course, but an RGB image cannot tell you much about the individual concentration of your fluorophores, and therefore it cannot relate to your object of interest. The second type, in this case multi-channel images, do allow you to do correlative or quantitative comparisons between multiple fluorophores or multiple objects, and should contain signal that directly correlates your isolated objects. As an example of a very basic file format, we have the regular Tiff file. These are usually a separate file for each frame or channel. However, they are also capable of storing multiple 2D, Z-planes or time series up into a single image with a maximum of up to 4 GB. There are many extensions to the Tiff files to try and circumvent this 4 GB limit, but they are not always as standardized, meaning that they cannot always be fully read by all analysis softwares. An example of an initiative that aims to standardize the reading and writing of metadata on top of TIFFs is the Bioformats tool, developed by the Open Microscopy Environment Consortium. They also developed the Ome TIFF file format. This is a microscope image standard that describes how to read and write 5D datasets with information like what objective lens was used, the numerical aperture, the lens's refractive index, and even emission and excitation wavelengths and much more. These are all very important for creating a theoretical PSF for deconvolution in Huygens. Huygens can read these parameters automatically, and since version 22.10 we also write Ome TIFF files. Besides the Tiff, there's also more proprietary file formats from different microscope manufacturers, such as CZI, ND2, LIF, or OBF, just to name a few. These can also be container files, meaning that one single file can contain multiple 3D images of different regions or even different samples entirely. This is a very neat way of storing images, as you don't end up with a different image file for each slice, time frame or fluorophore, but instead you get one single image file with images of many different objects. Opening a container file works just as easy as any microscope image. You can simply drag it into Huygens to open it. But after doing so, it opens a selection box with the content of the file. It shows you the image names and dimensions for you to make an educated selection. Sometimes, if Huygens detects that images in your container files are already partially processed, they will be color-coded to show you which is the unprocessed image and therefore would yield the best result for deconvolution. Now, let's take a look at an example of how to open a microscope file in Huygens. We have a microscope file here on the desktop. It's an ICS file, and we can look at its properties to see how big it is. This is an image that is 33.7 megabytes large. So if we open it into Huygens by dragging and dropping, it is now loaded, and we can see the histogram. We can see that this has 4 channels, which means that there are 4 fluorophores present in this image, and those were all acquired by the microscope. Furthermore, we can check the microscopic parameters of this file. We see that they are all green, which means that the values were reported from file, or they have a check mark, which means that they were that their value was verified by a user before. So the ICS file that we have, the file format ICS really contains a lot of metadata of the image, which is also called the image cytometry standard, which is a file format that we as SVI maintain, and we recommend users to use. A lot of other programs can also work with this file, like ImageJ or Fiji. On the right here, we see that indeed this image was 33 megabytes large. We see information of the data type, which we will cover later, and also the dimensions. And for each of the channels, we can see that their excitation wavelength and their emission wavelength is stored in the image's metadata. So this gives us confidence that we have a good microscope metadata for this image that can be used to generate a theoretical point spread function during deconvolution. So let's now accept these values and go to the deconvolution express to see what kind of deconvolution result we can get. After waiting for the initialization, we can click deconvolution express, and the loading bar on the bottom here tells you how far along it is, and this is a quite small image, and it is already done. We could also see the VRAM was used, so that the graphics card was being used to process this image. And if we now click done, it will be exported to the main window, and we have side by side the normal image and the deconvolved image. We can see that the histogram looks a bit different. And if we take a look at the microscopic parameters for this image, they will not have changed, but we can see that the data type is different and that the size has increased. Now, why is that? And for this, we need to go into something called data types, bit depth, and dynamic range again. So let's move back to our slides again. Let's now first do a little recap. So when acquiring an image on the microscope, you end up with an image that contains the intensity information of how much light of each of your fluorophores was captured at each sample point in your 3D space. This intensity is linked to how many photons were counted, so it's always shown in positive whole numbers, or integers. You could choose to store this intensity data for basic 2D images as a simple text file or Excel comma-separated value table, and then humans can read it. But this turns out to be terribly inefficient in terms of storage space. So efficient file writers save as binary data, and sometimes even do compression. But then you still have the flexibility in how much information you want to store per pixel. Storing 1 bit, or on or off, is enough to save information for a region of interest or mask image for analysis. And this is called binary. And each extra bit that you add doubles the amount of numbers that you can store in the image. There are a few standards in how many bits you want to use per pixel, and the end result is that we have different standardized data types. Using smaller data types uses less bits per pixel and saves in storage space. However, larger data types use more bits per pixel and increase the contrast and dynamic range of your image. The ratio between the highest and lowest intensity is able to be represented in your image is called the dynamic range. As a rule of thumb, if your intensities are within the range of 0 to 255, you can safely use 8-bit integer data type without the loss of any of your source data. This is the byte data type in Huygens. If your intensities are within the range of 0 to a little over 65,000, you can then safely use 16-bit integers without loss of data. But if you want to do some math for a specific analysis with these images, it might be necessary that you need to subtract images from each other. This could mean you end up with pixels where you would like to store them as negative numbers. And this is not possible in the unsigned integer case. For this, there is the data type called signed integers. The intensities are shifted so that we can have negative numbers, but as a result, the intensity range is halved because one half is used for negative numbers and the other half is used for positive numbers. For 16-bit signed integers, this means that we have a dynamic range from minus 32,000 up to plus 32,000. Note that so far we've only mentioned operations on images dealing with whole numbers. However, deconvolution requires the use of a PSF image. The PSF, or point spread function, describes your microscope's image formation process. It describes how your microscope blurs a single point of light after it has traveled through the optical system, and how it ends up onto your camera or detector. The light originating from your object is effectively distributed and smeared over neighboring pixels in XY and over different Z planes. In deconvolution, we aim to undo this blurring operation, thereby going from an image to a better representation of your actual object by using the information of both the acquired image and the point spread function. Of course, this is a simplified explanation because there are also many other factors such as noise, crosstalk, and aberration that need to be handled during the image restoration in Huygens. But the key takeaway for now is the PSF image. The sum of all the intensities in the PSF image is 1. So every pixel in this image has to have a value between 0 and 1. This is done so that the convolution and deconvolution operations obey conservation of total energy. This does mean, however, that a PSF image cannot be represented with whole numbers or integer values. So for the PSF, we need to use a different data type, one that can handle numbers with decimal notation. And computers deal with decimal numbers with so-called floating point numbers. These are decimal numbers that require 32 bits per pixel, which takes up twice as much memory and storage space as the 16-bit integer images. After deconvolution, the intensities in your original image will also be more concentrated towards their most likely place of origin, making the object itself brighter. The highest value in your image could then well exceed the 65,000 intensity limit for 16-bit integer images. So for these reasons, the deconvolution result is also always stored in 32-bit floating point numbers. Even though the intensity at a single point in your image may be higher after deconvolution, the total intensity in the image will remain the same due to conservation of energy. If your files are very large, which is the case for images that are used in stitching and even Lightsheet and Fusion workflows, you have to have a lot of storage space to store all the original images. You might be able to save those in an 8-bit format to save space if the intensities do not exceed 255. But if you also want to store the experimentally distilled point spread function in 32-bit floating point decimal numbers, together with the deconvolved and fused images in 32-bit decimal numbers for further analysis, then the resulting images will become more than 4 times larger than the original ones. For this purpose, file compression can be very useful. Here we see a very basic example of run length compression. If we have an original text with these letters, we can represent the data in a slightly different way, where the original text can still be recovered from the compressed representation without loss of any original data. It is important that when you do consider file compression, that you choose for such a lossless compression, so that the original information can be recovered one-to-one from the compressed information. There also exist lossy compressions, which is used in JPEG, for instance. As an example, opening JPEG and resaving JPEG files multiple times will pile up compression onto compression onto compression, leaving you with artifacts in your image. And these can then in turn also influence your deconvolution results. Our HDF5 file format has the ability to save files with lossless compression at the cost of taking a little longer to load and save the file when loading or saving in Huygens. Let's now take a look at a demo for file conversion and file saving in Huygens. So here we are back again with our previous example. Here we have our original image and our deconvolved image. Let's take a look at the microscopic parameters of this image. You can see that it is a 32-bit floating point image of 67.5 megabytes. 32-bit floating point means that it has decimal numbers, but also that it can exceed the 16-bit integer limit of 65,000. So this means that we can no longer save this image as an 8-bit integer image, and as you can see from the histogram here, the intensities go well over the 255 limit of 8-bit integers. Now, what would happen if we still were to save this image as a 8-bit integer TIFF? So we go on the top left to File, Save As, and then TIFF 8-bit. Well, there are multiple options that you can now choose from. First, they ask if you want to save it as one TIFF image per channel and slice, or only one per channel. Well, it would be nice to have a little less files. So one per channel would give only 4 files because we have 4 channels in this image. And we can save it on the desktop right away. So if you click Save, now we are asked how we want to convert this, because it's a floating point image and it needs to be converted to an 8-bit integer image. We have contrast stretch. This means that all the channels are individually scaled down in intensity to make sure they fit between 0 and 255. Each channel gets their own scale factor, and the results get rounded down to whole numbers, to integers. Linked scaling means that the entire image gets scaled so that their maximum intensity value fits between the 0 and 255. And clipping means no scaling is applied. This is okay if your image already fits between 0 and 255, but otherwise the intensity values that are larger than 255 will be clipped and put at its maximum value of 255. So we do not want this for our 8-bit TIFF, because our intensities lie well above our 255 limit. If we click contrast stretching and go to the task report, we see the values that each of our image channel was multiplied by in order to get the new intensities. So let's take a look at what this looks like if we load it back into Huygens. And to do that, for a Tiff series like this, for multiple channels, we only have to select the first one and drag it into Huygens, or go to File, Open, and then Desktop, and only select the first channel TIFF, not all of them at once, and click on Open. It will show the file series tool as mentioned before, and here it's noticed that this is a channel, and we can say we want to load channel 0, 2, 3, so that means all 4, or we can only read channel 0 and 1, but let's load all of them at once and then click on Load Selection. You could also load single file to load only the 0th channel, but we want to load the selection of all the channels. Then Huygens shows a message that the image parameters were not correctly found, because in TIFF files there are no image parameters saved regularly. So that's why the color is different. The information such as the excitation and emission wavelength are completely not found in the image. The only thing that can be found is the sample sizes in X and Y and the Z file sample size. Now, if we look at the histogram, we see that the intensities are approximately similarly scaled, but they all fit between 0 and 255. So the maximum value of each channel is now 255. but the relative intensity ratios are not kept in this way. So to do that, we had to select linked scaling, but that would mean that the green channel over here would get a lot less pixel gray values to be represented in than the rest of the channels, because it extends a little less. So then it would have only had 50 intensity values compared to the 255 it has now. So by looking back at the scaling factors, we know what the maximum intensity values of these channels originally should have been, and we can work it back and get it into our image. But we'd first have to convert it to a data type which allows for more intensity values than 255. So let's remove this image. To completely save all the metadata from the files, I'd recommend saving it in ICS or in HDF5. HDF5 has the benefit that it can compress down the image file. So if I save it as ICS on our desktop and then right click on the properties of this image, we see that it is indeed also 67.5 megabytes, just as the image is in Huygens. If we save it as HDF5, then after the file appears on our desktop, we right click and do properties again. We can see that the file size is also less. It's almost halved, and that is because of the compression. And this is lossless compression, so no actual data is lost. So when we read it in, it's the exact same histogram, it's the exact same data. Now let's say you have a lot of files, a lot of original TIFFs which you would like to convert to ICS. Then what you could use is the workflow processor. The workflow processor allows you to create workflows to do automatic deconvolution on very similar experiments, maybe do stitching, but in this case we would like to convert all our images from our input. We do not need to set the microscopic parameters, we do not need to set the deconvolution parameters. We simply want to select images, go to add, and maybe select our original ICS file. Then set save location, and by default it's saved to my desktop, but let's say I want to save it in my documents, and then here we can set we want to save it as HDF5, so it goes from our original ICS file to HDF5 file. And then we can click verify and next, send and close, and now we have a workflow that opens an ICS and simply saves it as HDF5, and we can run this. And if we now go to our documents folder, after the workflow has proceeded, we can see indeed that there is an HDF5 file which was saved and a batch file which describes what exactly the workflow has done, so we can reuse it later. And we can obviously apply this workflow to many of our input files at the same time. Finally, I would like to show you a way that you can also save TIFF data, but with Ome XML metadata. That way it stores the original image into a single file. You can also save one per channel or one per channel and slice, like the standard TIFF writer. But in this case, if we click a single file and click on our desktop again, it is saved as an Ome TIFF. And the Ome TIFF can handle 32-bit floating point numbers. And if we click on properties, it is now a little larger than 67.5 megabytes, because there is a little bit of extra storage necessary for the Ome XML metadata. But it is a couple of fractions of a megabyte, so this doesn't really add to your file that much. And we can simply drag and drop this into Huygens, and we can see exactly the same file, exactly the same histogram as our original file, and all of the metadata is saved. So the Ome TIFF is a very useful tool to have. And as a final example, I would like to open a problematic Tiff series, which I've saved on my desktop. I simply click again on the first TIFF, and it notices that it is a Z slice, which has 99 files in total, from 0 to 98. We click again on load selection, and again no image parameters have been saved with a regular Tiff, so we click OK, and here we see a cell division. It seems to be a regular image, but if we look from the side, we see weird artifacts, as if there is extra space between the layers. So let's investigate this a bit better in our slicer tool. If we now move from plane to plane, we see that there's a pattern that repeats every 3 planes. So this indicates to me that we might actually be dealing with a multi-channel image, which is wrongly read in as Z-planes, which should have been read in as channels. So it's not ideal, but we can convert this, we can fix it. So let's take a look at our original image. It's now 99 planes, but we know we have 3 channels. So here we see X, Y, channel Z between brackets, which means that the channel and the Z dimensions are interleaved, and we would like to pull them apart. And we know it has 3 channels, 99 planes in total, so our correct Z dimension would be 33, which is 99 Z planes divided by 3 channels. So now we have 33 Z planes per channel, and if we click on run, now we see that we have a more correct looking image. And if we were to have the microscopic parameters in a file, we could have loaded them here and have the correct microscopic parameters. But I do not have those with me right now. But we can look at the slicer again, and we see that with each plane I go through, I do not have this weird repeating and changing pattern. So if we look at it in a global color scale, we can see that each fluorophore is marked appropriately and is neatly separated. So that was all the information I have for today. If you have any more questions, please don't hesitate to ask them at info@svi.nl, or play around yourself, download the software, try out your batch file conversion yourself. You can see the link here to download our software. And that's it. Hope you have a pleasant day, and until the next webinar.